Good morning, everyone. You can hear me okay?
Yep.
Great. I hope you're all doing well, although I'm hearing a few of you coughing. I hope it's a mild case of cold and nothing else. So I'd like to ask you, if you don't plan to ask a question, please keep your microphone muted, and just to avoid the acoustic interference. That'll be great. All right, then. Aren't you glad to be doing this from home in this cold? Okay, do you have any, is everyone registered and ready to go on Connect? I've had a few emails since last class about issues registering for Connect, and I hope you're all good.
Yep.
Excellent. Do you have any questions for me before we begin? I take it that Alison Douglas is in communication with you as well regarding the lab. So I have not heard otherwise. So I believe things are going smoothly. Very good then. So today we will cover chapter 5. And so this will be the rate at which we cover the material for the course. Essentially, each class we meet, we will cover an entire chapter. Okay. Today's chapter is about the control of microbial growth. So we think of our world as two sections: internal, and that include yes. Okay, the internal compartments and the external compartments. That's our world. The internal compartment is our body, all internal tissues and organs, aside from mucosal membranes, our skin. Those two, as well as our environment, that's the outside world. Okay. So we try to control microbial growth in both of these compartments of our world. The internal compartment is covered with the antibiotics chapter, which we will talk about separately. Today we'll talk about control microbial growth in the other compartment, and that is the external compartment. So a little bit of a background. All of the techniques that we use today are derived from the microbiology work by the pioneers of the field, including, of course, Pasteur, as you know. Perhaps not as many of you are familiar with Joseph Lister. So in the late 19th century, Joseph Lister was a physician, a surgeon, and he struggled with patients undergoing surgery and contracting infections, internal infections. At the time, of course, the availability of antibiotics was very, very limited. So that created a large problem for physicians at the time. So Joseph Lister essentially experimented with different methods to try and minimize these infections, including the use of the so-called carbolic acid, aka phenol. So we know that today as phenol, and we use it regularly, in fact, in the laboratory. And basically, he tried to apply this material directly onto damaged tissue to prevent infections. Of course, he has some success. And the additional techniques that he suggested to employ, of course, the careful washing of hands of the surgeons during operations, although at the time he met with some resistance, as some physicians were not convinced that their hands could actually transmit infections between operations, between surgical operations. Nonetheless, the technique persisted, and it is used today, whereby the physicians wash their arms from the elbow down to their hands, as well as maintaining a clean environment in the surgical ward. And today, a lot of effort is put into maintaining a near sterile environment in surgical rooms. The importance of the contributions, of course, of Joseph Lester, we have it today. Some of you perhaps use the oral antiseptic Listerine that was named for him, of course, back in the late 19th century, when it was first introduced as a surgical antiseptic. Now, other oral antiseptics are available in the over the shelf in the drugstore, and they're all based on the same principle: the use of non-toxic material at appropriate concentrations, so as to remove and eliminate microorganisms without obviously introducing any toxic agents into the oral cavity. In addition to these basic techniques, of course, we have surgeons must use a mask. This is for their own protection as well as protection of the patient, because as a physician may exhale droplets, which can transmit viruses, of course, and bacteria onto the patient. So the use of a mask is mandatory. Eye covers as well. In fact, in surgical wards, even the paint on the wall is typically incorporates antimicrobial agents to minimize microbial contamination within those spaces. And in between operations, these wards, surgical wards, undergo strict procedures to clean up in between operations to maintain the sterility of that space. Let's talk about the principles of control. So here we will define a few terms, and they essentially reflect the degree of microbial elimination that is desired. You're familiar with sterilization. When we refer to material as being sterile, we mean that it is clear of all microorganisms and viruses. And that includes the highly resistant endospores, but does not consider prions here. Prions is the prion protein is a separate case, which we will talk about in one of the chapters separately. But it is a difficult protein to get rid of. Nonetheless, think of sterilization as the highest level of microbial elimination. And you're familiar with some of the methods used in sterilization. You've done some microbiology labs. The autoclaving is high pressure and high temperature is a method of sterilization, although there are others, and we will talk about those in this chapter. We have then the disinfection level. Disinfection refers to the process that eliminates most or all pathogens. So here we are focusing on pathogens and not all microbes. This is again a process whereby some viable microbes may remain, but this is a process used where the concern is only for microbial pathogens and not necessarily all microbes, because not all microbes are pathogens, as you know. Disinfectants are mostly chemicals that are used on inanimate objects, often called germicides. Bactericides are able to kill bacteria. If it's an agent that stops bacterial growth, it's called bacteriostatic, as you know. If it is specific for fungus, it's referred to as fungicide, virucides for viruses, and so on. So this is the process of disinfection. Then we have the process of antiseptic treatments. So these are chemicals used on living tissues specifically, such as cleaning the skin prior to an incision for surgery, whether it's an iodine texture or some other type of liquid agent that is used on skin or other living tissue. So this is specific for antiseptics. The next process is the decontamination, and this is essentially the lowest level of microbial control outside, of course, the body or the internal compartments. And this process is the use of washing or mild heat or chemicals to reduce the number of pathogens to a safe level. So it does not eliminate the pathogens, but reduces their numbers to a safe level. Then we have sanitization. Sanitization is basically what you would generally do around the house or the environmental workers do around the work space. Processes that reduce microbial population to meet accepted health standards that minimize spread of disease. So these include the wiping of surfaces, office desks, workspaces, etc., with wet wipes, the use of various agents to clean kitchens, bathrooms. Those are all sanitization procedures. We move then to the preservation process. And this is a process that is used to preserve perishable products. And it implies the delay of spoilage by microbes that are present in the material, in the perishable material. And here, you may also choose conditions of storage to slow the growth. For example, the use of a refrigerator in the kitchen is a method of preservation, essentially. You can also do the same by adding bacteriostatic agents, aka a preservative. So these are under the control of the FDA, in fact, because they are consumed along with the food material. So they need to be established agents as being non-toxic, at least to the level that is used in preserving the food material. And some of these are acids that are typically organic acids, really have no harmful effects at low concentrations at all. And finally, the process named after Louis Pasteur, pasteurization. And this is where the material is subjected to a brief heating to reduce the number of spoilage organisms. And this was developed to process milk, and today is used, of course, to process certain juices such as apple juice, wine, beer, etc., because those consumables do have microbes in them, and over time they can spoil. So pasteurization was developed to specifically destroy pathogens originally, as I mentioned, in milk, because pathogens used to be transmitted through the consumption of milk. Back, of course, in the concern for this was particularly focused in the late 19th century, at the time of Louis Pasteur. So this is a process that destroys pathogens. Other microbes remain. So pasteurization is not sterilization. The material will contain other microbes, although non-pathogens. The selectivity here, how this specific brief heating arrived at, is the fact that pathogens are generally more sensitive to higher temperatures than non-pathogens. There are exceptions, of course, to this rule, such as Clostridium, which is able to produce, of course, endospores. The temperatures that are used for pasteurizations are not sufficient to eliminate the endospores themselves. Although the vegetative cell, the vegetative microbe itself, is sensitive to those temperatures. So typically pasteurization, exposure of the liquid to temperatures between 55 and 72 degrees Celsius for specific time periods. And that is all that is used. And the idea there, of course, is to maintain the characteristics of the product. Okay, because if you boil this food material, particularly milk, and then try to repack it and put it in the refrigerator, it's going to look very unattractive, and it will change the taste as well. So those changes are avoided. So in selecting which method to use of the previous procedures that we've described, there are situational considerations, obviously. The purpose of the process, is it sterilization that you want to achieve? Is it degermination, etc.? And other situations, for example, is it food material? Do you want to preserve the texture of the food? Do you want to preserve the taste, the color? If we're talking about water treatment plants, of course, there are different considerations, obviously, used. And if we're talking about the laboratory or the medical setting, then we typically look at things like sterilization and antiseptics. Whereas in the case of water treatments, we want to remove as much of the microbial contamination as possible, but we do not really aspire to achieve sterilization of recycled water. All right, in terms of our daily life, of course, you've think you have a lot of experience in the past 2 years with COVID-19. So we have been using these techniques on a daily basis. The frequent washing and scrubbing with soaps and detergents, of course, for routine control. The rubbing itself in the presence of soap or even just water will mechanically help in removing microbes. The presence of the soap itself, of course, is toxic to the microbes, but even with water and the physical rubbing of the hands will mechanically remove microbes from the surface of the hands. Now, at risk here, there is the beneficial skin microbiota, which plays a protective role on our skin and inside our bodies. They are affected to a certain level, although they do tend to reside deeper within the skin and the hair follicles. Their numbers might be reduced, but they're not eliminated in these processes. Other methods that we use in our daily life, things like cooking the food, in addition, of course, to make it more appealing for eating. Example, if you're cooking meat, cooked meat would be more appealing to eat than raw meat. Cleaning surfaces, kitchen counters, bathroom counters, etc., as well as refrigeration, as we mentioned earlier. In hospitals, controlling microbes, of course, is very important because of the ever-present risk of health-associated infections, HAIs, aka nosocomial infections. And now we continue, of course, to face challenges even today with all of the techniques that we have in eliminating nosocomial infections or HAIs. They remain to be a major source of infections during visits to healthcare facilities. And there are various reasons for that, and we will talk about it in due time. Some of the main reasons, of course, is the fact those who are visiting the hospital are already sick, so they're weakened to a certain degree, and it makes them more susceptible to infection. They may also undergo invasive surgery, which means it exposes their internal environment to essentially microbes that are present in the air. And also the mix of bodily secretions, respiratory droplets, and bodily excretions such as urine and fecal material that will also invariably mix with material used in the healthcare facilities, and that aids in the transmission of these infections. Okay, so no questions so far, then I will continue. Now, we are aware that the health of healthcare workers and personnel is also important. It's not only the patients. We looked at an image earlier whereby healthcare workers wear a mask and eye covers. And this is important because they are continuously exposed to potential infections during patient visits to the healthcare facilities. And we've seen this particularly in the past 2 years with this COVID-19 pandemic. So the healthcare workers typically use standard precautions, protocols that are in place in hospitals and other healthcare facilities. Okay, and these standard precautions were developed by Health Canada and the assistance from other healthcare organizations to minimize the exposure, of course, of both patients and healthcare workers to HAIs. If a patient is admitted with an epidemiologically important pathogen, a highly transmissible pathogen, for example, with high rate of mortality, then additional procedures might be put in place, such as transmission-based precautions. So these are, think of them as higher level of protective protocols than the standard precautions. And you would see these in specific wards in the hospital. And today, these are quite prominent in the hospital ward where COVID-19 patients are placed. As I mentioned earlier, the operating room is perhaps one of the most important areas where microbial control is required. I mentioned the preparation of the patient, the use of antiseptics on the skin prior to incisions for surgery, washing procedures for the physicians and the nurses that are in the room and any other personnel. In fact, full cover with gloves, mouth and nose cover, eye cover, and entire outfit is covered, and the hair. I mentioned the floor and the wall. Specific or special paint might be used, whereby certain agents are incorporated into the paint. The air in the room is separate than the air in the other rooms in the hospital. So the air is recycled and is cleaned by passing through filtration systems to remove any airborne pathogens. I just want to show. Right. And then we have the instruments that are used in the operating room. So those need to be processed, just like we process instruments in the research laboratory. Obviously, any instrument you use, it needs to be completely sterile. And we'll talk about methods to sterilize these implements, which are typically metal. Those that are plastic are typically are not reused, so they are single-use material, again to minimize contamination and transmission of infections. So those are single-use plastic instruments that are thrown away after each use. So in the micro lab, there's particularly higher risk of contamination or infections if the person is working with infectious agents, simply because of the fact that we would be working with large samples, concentrated cultures with billions and trillions of microbes, and how you handle the specimen or the cultures is important. If you, for example, try to swirl a flask that has, say, 20 milliliters of microbial culture, in those 20 milliliters, you'll have trillions of bacteria or viruses. If you swirl it vigorously and to the point where you get air bubbles in the culture media, that automatically means that at that point there are airborne microorganisms from that culture. And now, assuming, of course, the flask is open, then you'll have microbes in the air. So even that simple procedure of trying to mix a culture by swirling the flask, it needs to be done in a gentle way to avoid production of droplets in the air. So because of this reason, of course, there are additional procedures that are used in the microbiology laboratory. And this is required not only for the protection, of course, of personnel in the laboratory, but also your microbial sample, because maintaining a clean sample is crucial to your work. You cannot work with a contaminated sample because any result you will obtain from all of that work that you put in would be useless. So obviously, careful treatments before and after, cleaning bench space, sterilizing cultures. In the micro lab, we don't simply dispose of material after work in simple way of throwing them in the waste and the landfill. The material is not released from the laboratory, in fact, until it has been decontaminated. So if we're talking about solid material, such as gloves, any other implements that are used, plastics, those are sterilized in the autoclave before they are disposed of with regular trash. Liquids, we do not spill any liquids down the drain in the laboratory without being pre-treated to kill all microbes in those media. So this would include potentially the use of detergents, which are the safest to the environment. But other cases, we may use more harsh conditions depending on the level of resistance of the microbe in the liquid. We could potentially use chlorines and other strong chemicals before disposal. Does it make sense?
Yeah.
All right. Okay, so the facilities that work with microorganisms are classified into one of 4 levels as established by the Centers for Disease Control and Prevention of the US. So in Canada, we follow the same type of classification. So these are biosafety levels for working with microorganisms. The lowest level is the biosafety level 1, BSL 1. And this is essentially procedure work with microbes not known to cause disease in healthy people. Okay? To a certain degree, of course. I mean, if you end up drinking a flask, 200 milliliters of microbial culture here, this, as you can imagine, will not really apply in this case. But this is essentially the lowest level. So any workspace with minimal procedures in place, such as having a lockable door, restricted personnel access, and those spaces can be classified as BSL1. The next level is BSL2. This is where the personnel in that space work with moderate risk microbes that can cause disease, but have limited potential for transmission. Okay. So here again, we will have that space has to have specific protocols in place, specific design, not being accessible to the public, and so forth. You can find those procedures, in fact, in Health Canada. If you go through their website, you'll find specific details of the requirements for each of these classifications. The next level up is the BSL3, and this is where personnel might be working with pathogens that can cause serious or potentially fatal disease through inhalation specifically. Okay, so I could tell you at Laurentian University, we do not have a BSL3 facility. The highest we have is BSL2. In all of Ontario, there are 2 BSL3 facilities. One is at the University of Toronto, and one is a government facility in Etobicoke. And the highest level is BSL4, And this is where work involves the use of deadly pathogens, such as, say, the Marburg virus, right? That can cause hemorrhagic fever. It's quite fatal. So this is the highest level. And in all of Canada, as far as I know, I don't know if things have changed in the past few years, but in all of Canada, there are only 2 BSL-4 facilities work with these pathogens. Okay, so let's now go into the different approaches for the control of microbes. Let's look at food and food production facilities. Perishables. It's important, of course, for perishables to retain their quality longer when contaminating microbes are destroyed. I mean, that's an obvious conclusion. So there are different procedures for food material, whether it's canned food, whether it's fresh food, whether it's processed food. And we'll go through these individually. So heat treatment, whenever possible, is the most common and reliable method used. And I emphasize here, when possible. Again, because we want to avoid changing the color or the food or the texture or the taste of the food, right? If heat is not possible, then other conditions might be considered. For example, irradiation and high pressure could be used. We can use chemical additives. Again, the preservatives that we mentioned earlier. Food processing facilities, of course, because they are processing food and they want to minimize contamination of the food material so that they do not have to use extreme measures to eliminate those microbes and thereby changing the quality of the food. They try to minimize, to maintain, of course, clean facilities. And the cleanliness of these facilities is also monitored by healthcare authorities, by regular visits of these facilities, by officers that would go through the facility and monitor how clean the facility is and look at their protocols, the written protocols that they use and their practices. In terms of water treatment facilities, so we recycle water for use, obviously, and water goes through a lot by the time it leaves inhabitable areas. Okay, so it definitely gets contaminated to a very large degree, and its treatment to remove pathogen is crucial for us to be able to recycle that water. Traditionally, chlorine used to be the first choice because it is highly effective at low concentrations at killing microbes. It has some drawbacks. It can change, of course, the flavor of the water, but well, it doesn't. Well, water doesn't have flavor. It doesn't change the flavor, but it creates a flavor that is undesirable. But what is worse, in fact, for high levels of chlorine is that the way chlorine works, or it is effective because it interacts with components of the microbes. That's how it kills them. So whether it's nucleic acids, lipids, or particularly proteins. So it reacts with those components, and that's how it kills the microbes. So any other organic material present in the water, the chlorine will also react with and produce the so-called disinfection byproducts, or DBPs. And There's been extensive research onto the health effects of DBPs, and some of them have been linked to sort of long-term health risks. So whenever possible, the treatment of chlorine is also avoided. There are some organisms that are resistant to the chemical infectants at the levels that we use. Right? So here, we're not free to use chlorine or other chemical agents at any level we want. There are upper limits that are established by the government. So, for example, Cryptosporidium hominis, it's a prominent pathogen that causes diarrhea, and it can be more serious in infants and children. So there are regulations that require facilities to minimize DBPs and C. hominis contamination in the treated water, and these are used as guidelines for the minimum quality of the water before it is being released for consumption. The approaches are also of concern for the pharmaceutical industries, the cosmetics, deodorants, and so forth. Obviously, they need to be clean, although some that already have alcohol in them, which is used in fact, as a chemical agent to control microbial growth. Others do not, so they would use preservatives or different soaps to achieve the same objectives. So the selection of the antimicrobial procedure, which one you use, there are factors that you must consider. Okay, there isn't really the perfect method to use. We are restricted, as I mentioned earlier, we are restricted by the level of microbial elimination that we want to achieve, but also the property of the material that's being used, whether it can withstand treatment with heat or irradiation, etc. And then you need to also to consider the type and the number of microbes. Is the material heavily soiled? Because the level of microbes is very important to how long you would treat the material for, and I'll talk more about this shortly. And the type, obviously, is the microbe of the resistant type or not? Is it able to produce endospores or not? And so forth. The environmental conditions that are used, the risk of infection. Is this are the microbes pathogenic, highly pathogenic, transmissible? And finally, of course, the composition of the item itself. Is it solid? Is it a powder? Is your food material, flour, for example? You would consider the treatment of flour or starch or sugar differently than a canned soup or a piece of canned tuna. So in terms of types of microbes, there are, of course, those that are sensitive. There are those that are resistant on their own, such as Mycobacterium species, which I've talked about before, having this sort of waxy cell walls. Pseudomonas species are more resistant than other as well. Naked viruses that lack lipid envelopes. Then we have protozoan cysts and oocysts. These are typically resistant to disinfectants, and they are excreted in feces from those that are infected. And they can be, of course, they can cause disease if ingested. These can be destroyed by boiling, if boiling is an option, 100 degrees Celsius. And then we have, of course, some of the highly resistant forms, such as the bacterial endospores, where boiling or even using dry heat at 100 degrees Celsius is not sufficient to eliminate them. So the point that I alluded to a couple of minutes ago, how heavily contaminated is the material is an important consideration. Why? Because the way that microbes are killed is represented in this curve. If we plot the time at a given temperature on the x-axis versus the number of surviving cells, you can see that if you have a logarithmic scale on the y-axis, you'll get this straight line that decreases. And notice that the decrease in the number of microbes is a logarithmic decrease. So essentially, microbes are killed 90% each time period. This is the so-called decimal reduction time, or D value. So if you start with 10,000 microbes and you apply your treatment at a given time and temperature, you will reduce the microbes from 10,000 to 1,000, 1 log unit reduction. The next unit of time and temperature will reduce the number to 100, then down to 10, then 1, then 0.1, and so forth. So because of this, obviously, the initial number of microbes is quite crucial. Okay? If it's heavily soiled, you would need either more exposure time or you would need higher temperatures. In this case here, we're fixing the temperature and we are plotting the time on the x-axis. Okay. This is the decimal reduction time. Now, you can produce a similar curve to give you the so-called decimal reduction temperature. And in that particular setup, on the x-axis, instead of plotting time, you plot temperature. And you fix the time. So you would say, I would like to treat my sample for 10 minutes. And if you plot the temperatures that you will use for 10 minutes each, you will obtain a similar curve. And that will give you the decimal reduction temperature rather than the decimal reduction time. Okay, pretty straightforward. Okay, let's move on. In terms of environmental conditions, these we also have to take into consideration. Okay, so dirt, grease, body fluids can interfere with heat penetration, obviously, because they themselves will consume heat. Action of chemicals, it will also interfere with the action of chemicals. Because let's take chlorine again as an example. Chlorine reacts with organic material. And that's how it kills microbes. It reacts with their proteins and lipids and nucleic acids and other metabolites. So if there are organic material in your sample other than the bacteria, obviously these chemicals will also, or these organic material will effectively consume the chlorine that you're using, thereby reducing the effective concentration of chlorine, thereby protecting the microbe itself. So these need to be taken into consideration, obviously. Temperature and pH. are also two obvious factors that can influence the effectiveness of the method that you choose. Typically, higher temperatures work better, even if you're using a chemical agent. Chemical agents are more effective at higher temperatures. pH is also important. Chemicals as well tend to be more effective at acidic temperatures, even heat. Let's take an example: sodium hypochlorite, or the household bleach. It can kill M. tuberculosis at 55 degrees Celsius in half the time as it does at 50 degrees Celsius. So a small shift of temperature, 5 degrees up, reduces, in fact, or increases the effectiveness by 100%. Okay, let's look at the risk for infections. Medical instruments are classified into different categories, essentially 3 distinct categories, according to the risk of transmission of using those instruments. The ones that come in contact with body tissues, the internal components or internal tissues and organs, those are classified as critical items, and they must be sterile. These include, of course, things like needles, scalpels, any other implement that is used during surgery. The next one is the semi-critical category, and these are the instruments that come in contact with mucosal membranes, such as the oral cavity, but do not penetrate body tissues. So the implements used by the dentists fall in this category. So these also must be, of course, free of microorganisms and viruses. Some endospores may remain, but are often blocked by mucous membranes, so they do not penetrate the tissue. For that reason, the classification here is somewhat lower than that of the critical items. These can include things like an endoscope or endotracheal tubes. And the least category is the non-critical instruments. And so the surfaces, the contact unbroken skin only. Low risk of transmission, countertops, stethoscopes, blood pressure cuffs, and other implements that may be used on the outside of the body. In terms of the composition of an item, which we talked about several occasions already so far, of course, if it's a plastic material, you worry about using heat, obviously, and other materials, right? The plastic and other materials that is sensitive to heat, you would reconsider the use of heat or try to lower the temperatures. Irradiation provides actually a good alternative in some cases, but it does damage some types of plastic. And I'm sure you all noticed that if plastic is exposed to the elements outside in your backyard, winter after summer after winter, the plastic actually breaks down, becomes brittle. It doesn't break down, I don't mean degrade. It doesn't biodegrade, but it breaks down in its consistency, or it becomes brittle because of the radiation from the sun over the summer months. Moist heat, liquid chemical infectants, of course, cannot be used to treat moisture-sensitive material. So if we are, again, if we're trying to treat flour or starch or sugar, obviously we cannot use moist heat or liquid disinfectants in those cases. Okay, so before going into the description of the different methods, Let's take a 5 and come back for the next section. Okay. Let's continue. Right. So now we'll describe the different methods in some more details that are used for the control of microbes. Right. First, the physical methods. Treatments such as, for example, filtration, irradiation, and high pressure. These are the most common physical methods that are used to control microbial growth. Again, they may or may not achieve sterilization depending on the specific protocol that is used. These are reliable, relatively fast, simple, and relatively inexpensive, and also non-toxic. I know that some of you might be concerned with the use of irradiation on foods because of potential production of toxic material as a result of treatment with irradiation. But the food security agencies and Health Canada, of course, have done extensive work on the effect of these different treatments on the food material, and they've essentially, if the method is approved, it means that it's safe. If the method is used properly, of course, right? Right. So heat. Heat is of two types, right? We have moist heat, where there's water or steam is involved, such as the autoclave. And we have dry heat, when you cook food material in an oven, for example, okay? That's dry heat. And they do differ in their effectiveness. At lower temperatures, moist heat tends to be more effective. It works by, heat generally, works by irreversibly denaturing proteins. And protein denaturation, for those of you who are not familiar with the term, when a protein denatures, it means its secondary and tertiary and quaternary structures have been destroyed. Not the primary structure, right? Which is the amino acid sequence of the protein, essentially. So the other levels of structures, the secondary, tertiary, and quaternary structures, are destroyed, and that essentially destroys the protein. It becomes non-functional. And this damage is irreversible. The protein is unable to revert back when you remove the heat. The protein is unable to revert back to its original form. Furthermore, during the process of denaturation, because it exposes the hydrophobic domains of the protein that are typically within the interior of the folded protein. When these hydrophobic domains are exposed in an aquatic environment, of course, in an aqueous environment, I should say, they want to shield themselves from the water because they're hydrophobic. So they will tend to actually aggregate together to shield the hydrophobic domains from the water. And this aggregation will cause them to precipitate, which again makes it essentially impossible for them to revert back to their original form. So boiling material is a form, of course, of moist heat, which takes place at 100 degrees Celsius. It is effective, but does not achieve sterilization, as we know. You need to raise the temperatures beyond 100 degrees Celsius to achieve sterilization. Pasteurization, again, it destroys heat-sensitive pathogens and spoilage organisms, but it does not eliminate all microbes in the material. Okay. So in the old days, they used to use lower temperatures, of 56 to 65 degrees Celsius for 20 minutes or 15 minutes, sometimes less. But commercially, that is not effective. Today, they use the so-called high temperature short time method, whereby they raise the temperature up to 72 degrees Celsius. And you only need to expose the milk to that temperature for only about 15 seconds to achieve pasteurization. So what they do, they pass the milk through pipes. The pipes are surrounded by a wire with resistance, meaning that it gets hot, and the resistance is controlled such that the temperature is around 72 degrees Celsius. And the length of the pipe is such that the milk passes through the entire pipe in a period of time of 15 seconds. So it comes out the other side, already pasteurized and ready for packaging in containers. Ice cream. The advantage, of course, of ice cream is that it needs to remain frozen. So they typically expose it to very cold temperatures during storage, but during processing, before packaging and storage, essentially, they raise the temperature to about 82 degrees Celsius for 20 seconds. Then they go into the freezing stage. There is the so-called ultra-high temperature method, and this is used for shelf-stable boxed juices and milk, such as known as ultra-pasteurization. For milk, it's 140 degrees Celsius for a few seconds, then rapidly cooled. And it does affect, of course, some of the properties of the milk as opposed to the standard pasteurization we mentioned above. Sterilization using pressurized steam. Okay, this is a process where you can actually eliminate all microbial life, including endospores. Oh, the image here to the right shows you schematically how an autoclave works. I'm sure you've seen similar images before. It's essentially an enclosed chamber whereby the interior temperature is raised, and there is steam that is injected into that chamber so as to raise the pressure inside. So the temperature, as a result of the pressure, the temperature of the steam rises up to typically 121 degrees Celsius. And the pressure is up to 15 pounds per square inch. The minimum exposure time that we use in the labs is 15 minutes at those conditions. So typically the full cycle from the start to the end is about 25 minutes. We give the protocol 5 minutes for the temperature and the pressure to rise to those levels. And then after the 15 minutes of steady exposure at those levels, there are an additional 5 minutes as the pressure is released and the temperature is brought down to safe levels to, of course, handle the material inside, because it comes out hot. So of course, you don't want to open the door of the autoclave while the pressure is still at 15 pounds per square inch in those temperatures. That's quite dangerous. Depending on level of contamination and the volumes, you may also want to extend the time within the autoclave. Flash sterilization at higher temperatures but shorter time can also be used. And how do we determine those specific conditions? Of course, there are ways that we can test them quite easily. we will include a culture of a specific microbe with the samples. So those can be test microbes. So you'll have your material in there. You'll have a test microbe after treatment. So this test microbe could be a small culture, liquid culture, or it could be an agar plate that is streaked with the microbe. After the treatment, you take it out, and you put it back at 37 degrees Celsius and see if anything grows. It's a simple way to test the protocol. Destroying prions involves immersion in 1 molar sodium hydroxide during or before autoclaving. That's how resistant prion protein is. Even though it's a simple protein, but it's very difficult to inactivate. Autoclave, of course, to be effective, steam must enter the item, obviously, right? And displace the air. So you place the long, thin containers on their sides. You do not close containers tightly to allow for the exchange of the, to displace the air within the inside. You can see outside those blue, the blue covers on those material. There is tape where you put a name, date, for example. You can identify the material, but the tape is a special tape that has an indicator in it. So to the left in panel A, to the left there, I want to give you use a pointer. So here we have an autoclaved sample, and this is the sample before autoclaving. Notice the change to the tape itself. You have the appearance of these blue dark bars, diagonal dark bars. These indicate that the material has been autoclaved already. So you don't have to guess. Here we have the same. Okay, in panel B, you have indicator bars that can give you an idea of whether the sample has been treated or not. Okay, well, let's go to the commercial canning process. And these commercial operations to prepare this kind of food, they use a huge autoclave called retort. And the food, the cans actually go into this autoclave through a conveyor belt. They could be on a large skid. So we're talking about thousands of cans that can go in at a time. And the material can actually be, can continuously go through the process on the conveyor belt, coming on the other side already autoclaved. So the microorganism of most concern for the canning process is Clostridium. Clostridium botulinum, to be specific. We know that this organism produces highly resistant endospores. The process, the minimal process in the production of the canned food, needs to be able to reduce 1,1200 endospores to only 1, a process known as 12D process. Now, this was established because it is not normal or virtually impossible to have that many endospores. This is critical because endospores can germinate in the canned food after the cans, the temperature of the cans are cooled. And of course, they're stored on the shelf, on the store shelf for extended periods of time, which could allow the endospore to germinate. And the cells, the vegetative cell is also resistant to low acid anaerobic conditions. They're quite happily, in fact, at those conditions. And they can produce the botulinum toxin, assuming they have the gene for it. Canned food commercially is sterile, so you can consider it commercial, the food to be sterile. Endospores of some thermophiles may survive, right? You know, thermophiles are extremophiles, and they are named so because of their ability to resist high temperatures. In fact, above 100 degrees Celsius. Some of them can be happily multiplying above those temperatures. But luckily for us, the thermophiles are rarely disease-causing microorganisms. There is no concern, health concern at that point. So that was the moist heat. Now we can talk about the other form whereby water and steam are not involved. So the most extreme form of dry heat, of course, is incineration, whereby you oxidize the cell material completely. You reduce it to ashes. And this is quite effective, of course, at achieving a sterile sample, even though the sample is not there anymore, technically. So in the microlab, you've done this process before. Remember using the wire loop that you use to transfer cultures or to inoculate liquid cultures or the surface of agar plates. And we have, of course, a flame, an open flame. We pass the loop, the wire loop, through the flame between each treatment to avoid contamination. Now, this is a form of incineration. It is also used to destroy medical waste and contaminated animal carcasses, whether in experimental setups or otherwise. Very effective way. There are the hot air ovens that can also kill microbes by destroying cell components. And here again, by denaturing proteins. So heat will denature protein, whether it's dry or moist. The difference is to achieve the same results, you typically need higher levels of dry heat than you do in moist heat. That's the take-home message. Longer exposure time. Right. So the next physical method is the filtration, and then from there we'll move to irradiation. Filtration is the simple process of passing material through a filter, and this could be again liquid, or it could be air, right? So HEPA filters, high efficiency filters, are used to reduce number of microbes in the air. In liquids, you can actually achieve sterilization by passing material through filters with well-engineered pores. So in this schematic, if you take a look here, this is the surface of a filter. You have a single bacterium, and here you have the sizes of the pores. You can see the sizes of the pores within the filter for the liquid to pass through. So the bacteria gets trapped on the surface of the filter, and the liquid will simply pass through. You can also use a vacuum pump to assist in pulling the liquid through the filter, right? So here, the size of the pore can be controlled So the manufacturer actually have engineering techniques to control the size of the pore. The standard size that we use in the lab is a 0.2 micron pores. Too small, of course, for any bacteria to go through. Yeah, even for some large viruses, they can be retained. So these are the so-called membrane filters. It's a very thin membrane, typically around 1 millimeter or thinner. The depth filters, on the other hand, they use porous material, and this is the example here. The HEPA filter is a form of depth filter. So various material that's in a fibrous form. Cellulose is often used. That is packed together quite tightly to produce a thick filter. While the material goes through the filter, electrical charges will trap the cells within the fibers of the cellulose, and the liquid material goes through. These depth filters are not as effective as the membrane filters because the pore size between the fibers is not well controlled. And so you can use this process also to treat air, again with the high efficiency particulate air filter, the HEPA filter. These, depending on the depth of the filter, how thick the filter is, how many layers are used, they can be quite effective at removing microbes over 0.3 micrometers from the air. They are used in, obviously, surgical rooms, hospitals, as I mentioned in the previous slides, and we use them in the laboratories, in the research laboratories, in the biological safety cabinets, where we handle microbial cultures. Now, I want to mention here difference between biological safety cabinets and fume hoods. Right? You're familiar with fume hoods in chemistry. Now, those hoods are not the same as biological safety cabinets. Okay, they look similar. Okay, it's a cabinet, a metal cabinet, typically, with different buttons to turn the fan on and so forth. But effectively, the process through which these two instruments work are quite different. Okay. So the fume hood in the chemistry lab, there is of course a fan to create vacuum that will suck the air out of the hood up through different pipes, and are essentially the air passes through a flame, okay, to incinerate any chemicals that go through. The biological safety cabinets actually works in the opposite way. So the chemical hood has negative pressure inside. Right, as I mentioned, essentially the fan will create vacuum to suck the air out of the hood. So the air from the room will go into the hood and then get sucked up, along with any vapors of chemicals that are present in the hood. So this is a negative pressure. The biological safety cabinet has positive pressure, meaning that air is actually pushed in from outside into the hood. Okay. Now you may ask yourself, well, that means that any microbes are in the air, the hood is going to be pushed out of the hood into the person working in the hood, right? Which at first glance, you're right, that's what would happen. But so there are additional features to the cabinet which does not allow for this to happen. There are two things. One is typically there is a cover made up of either hardened plastic, plexiglass, or actual glass. the door is lowered to shield the person that's working there, and also to minimize the loss of the air from the hood as it is being pushed in from the outside. Okay. Now the second feature is that along the entire cabinet, where the person is using the elbows and the arm, okay, there are holes, and in that area there is in fact a negative pressure to suck in any air that comes out of the hood. Then it is recycled through a filter, a HEPA filter. That's the difference between these two forms of cabinets. Right. So the last physical method we're going to touch on is the irradiation. You're familiar, of course, with irradiation. We typically use the short wavelengths of the spectrum. So anything below, anything in the UV region and below, shorter wavelengths. So to be bactericidal here, now ultraviolet can be effective to a certain degree, and I'll talk about differences between ultraviolet and other forms of radiation. Then we can go shorter and go to X-rays, and we can go even shorter and go to gamma rays, which are more effective than both X-ray and ultraviolet radiation. So short wavelengths, high frequency, obviously has a lot more energy than low frequency, and it can also produce the so-called reactive species, reactive oxygen species, and other reactive molecules such as hydroxyl radicals. And those reactive chemical species are high energy, high speed species that last for a short period of time, but because of their high energy and high speed of travel, they will break any molecule they collide with, including double-stranded DNA. They can break them clean if they are in the immediate vicinity of that DNA. The same for proteins. They will damage proteins, lipids, and so forth. And that is really the secret of their effectiveness. So the high-frequency short wavelength radiation we call ionizing radiation, because they produce these reactive ions. Okay. They produce reactive oxygen species, reactive hydroxyl radicals, and they can do direct damage on molecules. And the radiation is used, obviously, to sterilize heat-sensitive material. Okay. Strawberries, of course, you cannot clean them or remove microbes from them by treating with heat, because you'll end up with cooked strawberries, and you don't want cooked strawberries, you want fresh strawberries. And this is typically used after packaging. So the material, after it's ready to be shipped, it passes through the ionizing radiation chamber before it comes out, and it's ready for shipping. Ultraviolet, the least effective of the three types of radiations, it can produce direct damage in DNA by forming dimers. It is used to destroy microbes in air, water, and you have to use it in a certain way, by the way, to be effective in water, and especially effective on surfaces. It does suffer from its poor penetration power, unlike X-rays and gamma rays. So it is typically used to expose surfaces, which will eliminate microbes on those surfaces. If you pass air through lamps of UV lamps, or if you pass water in thin films through UV lamps, you can achieve some forms, in fact, of microbial elimination. Of course, exposure to any of the radiations that we mentioned, including UV, is damaging to skin and eyes, so you avoid direct exposure to these radiations. Microwave ovens, they suffer from the fact that they do not heat food evenly. So you may actually have a particularly thick material like a bowl of soup, for example. It's quite viscous and thick, and we'll find that there are pockets within the soup whereby the temperature actually is not hot. So this uneven treatment makes microwave not suitable, unless, of course, the food material is in a pan in a thin film, and you expose the material to for a long enough time, and also in between you actually mix the material and then apply the microwave again. You can, in those cases, achieve good treatment. Oh no, there's the high pressure actually. This is the last one of the physical methods. So this is used also in the commercial operations, food preparation material, particularly in fruit juices and guacamole. High pressure up to 120,000 pounds per square inch. That's a lot of pressure, in fact. And it does so by denaturing proteins, just like in the case of heat. And it can also, of course, alter cell permeability, as the pressure will essentially burst the cell membrane and the cell wall. And in this particular treatment, the product can maintain its color and its flavor. But it is somewhat costly to use this kind of procedure. Okay, so let's take another 5 before we go into the chemical methods. Sorry for the delay. I rushed to make coffee and I spilled it. Sorry. I had to clean it up. Okay. Let me see. I have some. So okay, good. Let's chat between you guys. Oh, I'm good now, right?
Yep.
All right, excellent. Good. So let's carry on. Where were we? Chemical methods. Right. So the chemicals that we use to basically degerm or sanitize or sterilize different surfaces, including biological surfaces, like our skin and mucosal membranes, such as the oral cavity or any other mucosal membranes. Germicides are generally referred to as disinfectants, but in some cases, as we will discuss shortly, they can achieve sterilization. So chemicals have the advantage of reacting irreversibly with proteins, and I as an example, I talked about chlorine previously, and how effective it is, and the fact that it forms new chemical species when it reacts with various proteins, as well as metabolites. Any metabolites that react with and form different or new agents. So this irreversible reaction with proteins, nucleic acids, membranes, and of course viral envelopes are proteins, and membranes, or membranes rather, with proteins in them, which essentially is a cytoplasmic. It's very similar to a cytoplasmic membrane. Okay, viral envelope is very similar to a cell membrane, and we'll talk about how it actually acquires this structure, because viruses on their own do not make their envelopes. Okay, they simply steal it from the host after infection. So this irreversible reaction, regardless of the nature of the cellular component, it renders this component defective, right? So they are quite effective at neutralizing microbes. It is quite good for treating large surfaces and heat-sensitive items. Again, if the physical methods that we've described are not suitable for the specific application, it changes its properties or physical properties or flavor or color, etc., texture, then you may be able to use the chemical methods. And some are quite safe, right? Sufficiently non-toxic to be used as antiseptics. And some are used, in fact, as preservatives. And these are organic acids that we find even in our bodies. Not at the same concentrations, but they are organic acids nonetheless. Right. So again, in the United States, the Federal Drug Enforcement Agency is responsible for ensuring the chemicals used to treat medical devices, and of course they regulate drugs as well, because of the fact that these are chemicals. They're also regulated by the same organization. there are in fact hundreds, if not thousands, of different agents out there that are allowed in non-prescription antiseptic products. But there must be some kind of proof, experimental evidence, some type of research on the material to actually prove its effectiveness and its safety at the same time, right? So we have antiseptic washes, such as soaps. We have antiseptic rubs, such as the hand sanitizers, and these have been very popular the past couple of years because of COVID-19. We have topical antiseptics that are used, of course, in healthcare facilities, obviously, right? Disinfected the skin, patient skin, in preparation for injections or surgery, incisions. And there are antiseptics used, of course, in first aid products or by the food industry as well. Now, selecting the appropriate germicidal chemical, of course, here again, you need to take into consideration certain factors. The toxicity level must be weighed against the risk of the use, activity in the presence of organic material. Again, because it's a chemical, just like in the case of chlorine. Presence of organic material other than the microbes themselves will interfere with the process because it will end up consuming some of this chemical during the process, thereby reducing the effective concentration of that chemical agent. So if there is heavy organic contamination of your sample, other than the bacteria, whether it's foodstuff or otherwise, it means you need to increase the concentration of your agent, of your chemical agent. Compatibility with the material being treated, it's an obvious one as well. Obviously, you cannot use liquids to use on electrical equipment because you will have a bad result and a bad experience with electrocution or, of course, a short in your house or a fire, potentially a fire as well. Residues that are left, let's say we're treating a solid surface, a countertop, etc. The treatment will invariably leave some kind of a residue on the surface. And this residue, depending on the nature of the chemical agent you're using, it can be toxic if you were to use the surface for food preparation. And it can be corrosive and actually damage the surface itself if left there for extended periods of time. Cost and availability, storage and stability are secondary factors that one may take into consideration. Concentrated stock decreases, of course, storage space because every time you need to use it, you dilute it with water, right? You need to consider environmental risk as well, right? Because the stuff will eventually find its way out through the drain or the landfill, and with the rainwater, that material will find its way into the groundwater system, or through the streams and rivers into the lakes, and eventually into our immediate environment. So environmental risk needs to also be taken into consideration, obviously. Now, in terms of effectiveness of the chemical agents, again, they're also classified into distinct categories. So we have the sterilants that destroy all microbes, also called sporocytes, because they're able to sterilize, means to really remove spores as well, endospores. So they're also sporocytes. And these are quite effective for heat-sensitive critical instruments. For example, instruments that contain plastics and they need to be used in sterile tissue. Obviously, if they have plastics, you cannot use the heat, right? So you can use these sterilants quite effectively. Next level, we have the high-level disinfectants that destroy viruses, vegetative cells, but not reliable to remove endospores. So this would be the next level down, right? And these are used on semi-critical instruments. Again, the semi-critical instruments, the ones that come in contact with mucosal surfaces, again, as in the dentist office, but not that come in contact with the internal tissues and organs. Then we have the next level down, we have the intermediate level disinfectants that destroy vegetative bacteria, mycobacteria, fungi, and most viruses. And these are used on non-critical instruments. And lastly, we come to the low-level disinfectants that can destroy fungi, vegetative bacteria, except mycobacteria and envelope viruses. Obviously, these do not eliminate endospores and non-envelope viruses. And these are used for general cleaning, such as disinfecting furniture, floors, walls, countertops, and other surfaces. The different classes of chemicals used: germicidal agents, the alcohols. We're all familiar with alcohol, aside from drinking. We use it for as germicidal agents. Of course, a good number of germicides that are used, including hand rubs, contain alcohol. The preference is typically isopropyl alcohol, seconded by ethyl alcohol in terms of preference. And it's used at a concentration between 60 and 80%. Most often, what we use in the lab is, in fact, a 70% concentration. Why would we use 100%? It'll be more effective, no? Anyone want to take a crack at answering that question?
It's toxic and it would evaporate very quickly.
Yes, both good points. It does actually evaporate quite quickly. So it may evaporate before killing the microbes, although it may denature some of the surface components of the microbes, so it does not penetrate. It doesn't stay long enough to penetrate the cell and actually kill it. And the fact that it's more toxic at 100% than 70%. Right. So alcohol preparations are not reliable against endospores and non-enveloped viruses. So they're not typically considered to be sterilants, right? It works by denaturing proteins and solubilizing lipids, membrane lipids. And that would be, of course, enough to kill vegetative cells. Because proteins, alcohol essentially, the denaturing process, as I mentioned earlier, whether it's with heat or whether it's with alcohol or any other chemical, the denaturing process will expose hydrophobic domains, irreversibly damages the protein and causes them to aggregate and precipitate. Both of these results contribute to the effectiveness of these agents. The alcohol is typically used as antiseptics and disinfectants at the lower concentrations. They're non-toxic, not on the outside, of course. Okay, you don't go drinking the commercial 70% alcohol preparations, right? Even if it's ethanol. Okay, that's very toxic. because of many reasons, but particularly it may contain high levels of methanol, which can make you blind. But on the surface, we're talking about, of course, non-toxicity to the surface. So coming in contact with your skin is not at those levels. It's non-toxic. It's fairly cheap as well, and because it evaporates, it does not leave any residue. So some of the advantages of using the alcohols. Its limitations, of course, it evaporates quickly, limiting the contact time, but we can get around that. We can use it in heavier loads, sort of increase the amount of alcohol and reduce its concentration from pure alcohol to about 60 to 80%. Other things to consider when using plastic, it does with extended use on the same surface. If it's rubber or plastic, the repeated use may actually damage those surfaces. If it's of concern to you. The preparation of tinctures, such as iodine tinctures, are essentially iodine that is dissolved in alcohol, as opposed to simply iodine. Next class are the aldehydes. Well, the aldehydes are related to alcohols, of course. It's an oxidized form of alcohol. Two common forms of aldehydes are used: the glutaraldehyde and formaldehyde. There is a third agent that is not as commonly used, called orthophthalaldehyde, or OPA for short. But the formaldehyde and glutaraldehyde are more commonly used. They work in a similar way to alcohol, with the added effect of cross-linking proteins. So in addition to denaturation, these agents can actually cross-link proteins together, and it's a feature that will cause them to aggregate and rendering them inactive. Okay. They are considered more toxic than alcohols, however. They're commonly used at a concentration of about 2% in an alkaline solution. That's the form they're typically used in. And for treatment of medical items, medical implements, the implement is simply immersed for up to 12 or longer hours. And again, they do suffer from the limitation of them being actually more toxic than alcohol, which means that you need to thoroughly rinse them after being immersed for 10 to 12 hours. The OPA is less toxic than both glutaraldehyde and formaldehyde, but it does change the skin color. Okay, an obvious disadvantage, turns the skin gray. Formaldehyde can be used as a gas, as formalin, 30% solution. It is effective against most microbes. It kills bacteria and it can inactivate viruses. In fact, for vaccines, this is one way. Now you know that vaccines sometimes vaccines can be components of microbes, so they can be proteins. They can be mRNA. In the case of the COVID vaccine, of course, it's an mRNA. But all previous vaccines, if it's a component, it is typically one or several proteins together from a pathogen. Okay, a vaccine can also be an entire microbe, an entire microorganism, and that microorganism needs to be dead, or you cannot, of course, inject somebody with a pathogen as a vaccine, a live pathogen. for obvious reasons. Basically, the pathogen would be inactivated by treatment with one of these agents, and then dried, packaged in a vial, and then shipped for treatment. At the healthcare facility, it's reconstituted from the solid material that was air dried, or not air dried, freeze dried, lyophilized. It will be reconstituted with a saline solution before injection. So these treatments will entail use of the formaldehyde as an example. There are, of course, live vaccines that are used. The Bacillus calmette-guerin, or the BCG used for TB, is in fact a live vaccine. Is a bacteria that is live, but it's not, of course, a virulent form of the bacteria. It is used to preserve specimens. So a large number of specimens in biology are stored in aldehydes. Obviously, another serious disadvantage is its implication as a potential carcinogen, the aldehydes. The biguanides, such as the chlorhexidine, and you probably seen this chlorhexidine if you ever read components of various off-the-shelf material in the pharmacy, whether it is hand scrubs or similar liquids. They may have chlorhexidine as the active ingredient. It's used extensively as antiseptic. It can leave residue and stays on the skin and mucosal membranes. So they can be used in the oral cavity under certain conditions, of course, and at certain levels, because of its relatively low toxicity. It is capable of destroying vegetative bacteria, fungi, and some envelope viruses. Remember, the naked viruses are the more resistant form of viruses, okay? Or the non-enveloped, aka naked viruses. We find that used in skin creams and prescription mouthwashes, chlorhexidine. Also in some preparations in the hospitals. Next one is ethylene oxide. Ethylene oxide is a gaseous chemical, and it's quite effective. It is in fact a sterilant, and it can be used to sterilize heat or moisture sensitive items. It does destroy endospores, which means it can destroy all other forms of microbes, because the endospores are the more resistant form. It has the advantages of being able to penetrate fabrics, equipment, and implantable devices, such as catheters. It's a quite important item to treat. Mattresses, electrical equipment, artificial hips, and other implantable devices. It's also used for many disposable lab items, such as petri dishes and pipettes. That's how they're sterilized before they're shipped for consumption, for use. It's applied in a special chamber, a lot like the autoclave, okay, where you will have temperature steam, and you'll have the ethylene oxide in the gas as well. Its limitations, minor limitations of being explosive. So, however, the end user at the concentrations that we use in the labs, explosion risk is low. It is mainly a concern for those that are manufacturing the product because it is present in high concentrations, in concentrated forms. It is obviously, if it's explosive, it will invariably be toxic. I don't know of any safe agent that is explosive. It's safe to consume. Also potentially carcinogenic. Few minor limitations, although it is quite effective and it's still in use. Must be eliminated by heated forced air for 8 to 12 hours. So there's the way to get around all these limitations if it must be used. Okay? By heated forced air over time. Halogens, right? Halogens are oxidizing agents. They also work by reacting with proteins and activating them. So essentially, they form chelates, particularly around charged amino acids on the surface of proteins, phospholipids, nucleic acids, and they render them inactive at that point. Chlorine, as mentioned, quite effective. It is capable of destroying endospores and all other forms of microbes. It is caustic to skin and mucosal membranes, so you want to avoid high concentrations. Obviously, we know Javex, aside from the fact that it stinks if you use it around the house or in the lab. In the lab, you may be able to handle it because it's quite effective and there are large or heavily soiled surfaces with large numbers of microbes, whereas at home, some may not actually be willing to tolerate the corrosive effect of Javex or Chlorix or its strong smell. Typically in the house bleach solutions, it is diluted 1 to 100 times before it is being, when it's used. It is very low levels, disinfecting drinking water to get rid of Cryptosporidium oocysts, as well as Giardia cysts. Cryptosporidium is of concern, as we mentioned earlier, even Cryptosporidium hominis. But the oocyst and of Cryptosporidium and the cyst of Giardia are of particular interest, and they can be effectively removed with diluted Chlorix solution. Can react with organic compounds in water, just as we mentioned earlier. So presence of organic material other than the microbes in the liquid need to be taken into consideration because it will self-consume some of the chlorine present, thereby reducing the effective chlorine available to attack the microbe. It can produce potential carcinogens depending on which chemical, which organic material is present and which chemical reactions will be catalyzed. Some of these products can be carcinogenic. Chlorine dioxide is used as a disinfectant and sterilant, so in the form of gas. It's quite effective, in fact, as an oxidizing agent. The second halide or halogen is the iodine. Iodine works very similar in a similar way to chlorine, but it is not as effective. Therefore, if endospores are of concern, typically chlorine is the choice, not iodine. But iodine is considered less toxic than chlorine, and there is no known potential carcinogens produced as a result of reactions of iodine. So it is the more commonly used form of tincture in alcohol at different percentages, and these have been used before on the skin in preparations or to treat wounds. It can also be used as an iodophore so in the absence of alcohol, whereby the iodine itself is slowly released from a carrier molecule. It is less irritating and longer lasting than tinctures. Pseudomonas, keep in mind, can survive in stock solutions of iodine. That's how bad Pseudomonas is. Metal components, okay, they combine with self-hydryl groups of proteins, so cysteines and methionines. High concentrations are considered too toxic to be used medically. Silver, it's used in some occasions in some creams and bandages because it is relatively cheap. Of course, while we don't go to using items or things like mercury or lead, even though they might be effective, they are quite toxic, but not so much for silver. Silver nitrate, eye drops, were used for over 100 years to put in the eyes of newborns as eye drops, because newborns used to be quite susceptible to eye infections, which can result in blindness. Silver nitrate eye drops used to be very effective in eliminating those infections, particularly due to Neisseria gonorrhea. But today, they simply use paste, antibiotics in the form of paste or eye drops. If it's a paste, they simply rub the outside of the eye, the eyelid, both eyelids with that paste, or use few drops of standard antibiotics that we have to replace the silver nitrate. Compounds, of course, of mercury, tin, copper, and others can be used as preservatives, but their setbacks and their limitations are obvious. They can build up in our bodies, and their building up is quite toxic and can contribute to several diseases, including fatal ones. So now they're strictly regulated in terms of use. And next we have ozone, O3. Ozone, we know that we've heard, of course, of the ozone layer. Ozone is produced naturally in the upper atmosphere as a result of interaction of radiation from the sun and oxygen. It is considered an unstable form. It decomposes quickly, so it's typically generated on site when it's to be used. So it's not shipped in containers or vessels. The instrument would simply be some kind of a vacuum that will take in air from the room, and inside it will be exposed to electrical plates, and that electricity will actually generate the ozone, which comes out of the other side to treat specific surfaces. It's quite powerful as an oxidizing agent, and in some cases it is used as alternative to chlorine or drinking water and wastewater. I know some household machines are available commercially, where people put them on their night table or beside the table lamp. They produce low levels. Now, ozone is toxic, obviously, because it is capable of killing microbes. It's also toxic to us. But some propose to use these, again, in these household machines to produce low levels of ozone, to clean the air from microbes that are attached to dust floating around in the air. But this is quite risky, and those machines are highly regulated. Peroxygens, okay? Another form of powerful oxidizers aside from chlorines and ozone, and these are powerful enough to be considered sterilants. They are readily biodegradable, so they leave no residues, less toxic than ethylene oxide or the aldehydes. Hydrogen peroxide is an example, quite effective, and the effectiveness depends on the surface itself. Okay, aerobic cells produce enzymes that are capable of neutralizing hydrogen peroxide. So whether we're talking about aerobic microorganisms or we're talking about our own cells that are aerobic, okay, so they can break down the peroxide with enzymes like catalase. There's superoxide dismutase as well in some microorganisms, and in fact is one of the tests that are used to test quickly if a microorganism is aerobic. Okay, you put a drop of peroxide, and if gas bubbles appear due to the release of oxygen, of course, you know that microorganism produces that enzyme, which means it is aerobic, because the anaerobes are not normally exposed to oxygen, and they have evolved not having that enzyme because there's no need for it. Peroxide is more effective on inanimate objects. It doesn't damage most material. It doesn't leave any residue. Hot solutions or vapor-based are very, very strong, and they are used in the form of sterilants. Peracetic acid, another peroxygen. It is even more potent than peroxide. It sterilizes in under an hour, and it's effective even in the presence of organic compounds. Like peroxide, doesn't leave residues, and it is widely used on a wide range of materials. Phenolic compounds, such as phenol, we've touched on early in the class. It's one of the earliest disinfectants, in fact, but it does have bad odor, and it is quite irritating to the skin. And obviously, it is toxic for internal consumption, right? It is very effective at destroying cytoplasmic membrane and denatures proteins. It is one of the favored disinfectants used to treat surfaces where mycobacteria work is involved, like Mycobacterium tuberculosis. It is quite effective against vegetative bacteria, but not reliable on viruses. Okay? Reasonable cost remains effective in presence of detergents and organic contaminants. Some of the appealing features of phenol, and it can leave antimicrobial residue, which is good. It's a good thing. Hexachlorophene and triclosan. These you may have also come across if you read components of material that's on the shelf in the drugstore. They're also used in hospitals and other health facilities. FDA recently limited some use of triclosan, but the hexachlorophene enjoys wider use still today. So that brings us to the quaternary ammonium compounds, or quats for short. So these are cationic detergents, and they are referred to as quaternary ammonium compounds because they're composed of nitrogen. I'm not sure if they have it in the slides, but they have nitrogen. Now, nitrogen has how many bonds normally? Nitrogen typically has 3 bonds, right?
Yep.
Now, these ones, quaternary for having 4 bonds, actually around nitrogen, which gives it what, a positive charge, right? And these 4 bonds, of course, those side chains can be a variety of groups. Typically, they would have a ring system, a lot like the benzene ring system or fused ring system. Some may also have hydrophobic side chain, so several hydrocarbons, making it a lot like a detergent. Okay, so whether it's a hydrophobic ring system, such as benzene rings, or hydrocarbons, such as fatty acids, because of the hydrophobic nature of the side chains and the charged nature of the ring of the nitrogen, now we have an amphipathic molecule, which is the definition of a detergent molecule as an amphipathic molecule. It has the advantages, of course, of low toxicity. It's used quite extensively in restaurants and food preparation surfaces, even at home. It works by reducing the surface tension of liquids, just like any detergent. It's effective as a degermation agent to remove dirt, organic material, microorganisms, obviously, along with those as well. It reacts with membranes. Of course, being amphipathic means that as a detergent, it will penetrate membranes. It will solubilize lipids, thereby breaking up the cell barriers, of course, and contribute to destroying the vegetative bacteria and envelope viruses. Now, naked viruses do not have envelopes. That's why they're called like that, which means that the detergents or quats will not really have much effect on the surface of the virus, which would be mainly protein in nature, in the absence of lipids. Pseudomonas, again, is able to resist the quat salt. So another nasty form of Pseudomonas. Preservation of perishable products. So chemical preservatives, I mentioned organic acids before, weak organic acids, right? So I'm sure you've seen these. Benzoic acid, used quite extensively, sorbic acid, even more, and propionic acids. Sorbic and propionic acids, those are considered safe organic acids, even at relatively higher concentrations. So these are typically the ones of choice. Benzoic acid can be used as a preservative in topical creams because it is not consumed with the food like the sorbic acid or propionic acid. Benzoic acid in topical creams can be used quite safely. For example, any creams to treat cold sores, for example, or skin irritations and so forth can have benzoic acid as preservatives. So these work again by affecting cell membrane function. They're used to control molds and bacteria in food, okay, such as sorbic acid and propionic acid. Citric acid, also another common acid, perhaps is one of the most commonly used weak organic acids to preserve food, citric acid. Nitrate and nitrite are used quite extensively in processed food. And you find these, they're quite effective as preservatives. And you'll find these in wine, preserving wine. You'll find them used on dried meat, right? Like pepperonis and those other dried meats that you can buy in the store. They do inhibit endospore germination and vegetative cell growth, the nitrate and the nitrite. Except that you don't want to eat a lot of those. You don't want to eat high amounts of these. They're quite effective at stopping the growth of C. botulinum. And they are used illegally to increase the or to enhance the color of meat. It gives it. I know some crooked merchants. They will reshelf expired meat after treating it with large amounts of nitrates and nitrite, because when they rub it on the meat, it recovers sort of the pink color of the meat, making it look fresh. Yeah, that's, of course, an illegal practice. When cooked, the disadvantage of these, the nitrates, if they're cooked, particularly if you're barbecuing with an open flame, they tend to convert to chemicals known as nitrosamines. And nitrosamines have been implicated for the past 50 years as potential carcinogens. Low temperature storage. This is a form of preservation, of course, again, because low temperature slows down metabolic activity and therefore eventually slows down microbial growth and prolong the shelf life of foods and pharmaceuticals, including drugs, right? Useful drugs. Okay. Now, of course, as we know, just like the extremophiles that we talked about earlier, thermophiles that live happily at temperatures beyond boiling temperatures, there are those that live quite happily below zero as well, right? The psychrotrophs and the psychrophiles. Luckily for us, again, just like in the case of the thermophiles, we rarely have pathogenic microorganisms from these two groups. Freezing also another form of preservation. Of course, it reduces the growth even further, essentially stopping it because the temperature is so low. But cycles of freezing and thawing are in fact a good way, are antimicrobial in that form, okay? Because especially if it's a liquid, not like a piece of meat, okay? Because the freezing, as you know, produces snow or ice nuclei, right? Because that's how ice forms. It starts as a nucleus and then it grows in size. And the way it grows is an irregular growth pattern, and it can produce sharp edges and points, sharp points as the ice increases in size. And essentially, if you have bacteria there, these growing edges of the ice can actually damage the bacteria. So if you, they can do the same, by the way, to protein preparations. So when we store enzymes in the lab for research, we store them. You don't store the entire stock in the freezer. Take it out, thaw it, take a very small volume, then put it back in the freezer. Then do that every time you want to take out some of it, okay? Because that will eventually damage the protein. So the better way, of course, to store material if you have to freeze it is to, as soon as you receive your stock, you fractionate it into smaller, manageable volumes that you may need. Okay, let's say you get a stock solution of 1 ml, but you normally use 50 microliters at a time. So you don't want to freeze and thaw that sample 20 times to use up the 1 ml. Okay, it's better that you actually fractionate it immediately into 20 tubes of 50 microliters each, and freezing those. Then when you need to use one, you simply take it out, thaw it, and use all of it. right? Okay, dehydration, or more generally, reduction of available water. Okay, dehydration is a form, but it's not the only form to reduce available water. If you use something that's very, very concentrated, you essentially reduce the amount of water that is there, right? So salting, when you add salt, you are reducing available water. If you add a lot of sugar, I know this sounds counterintuitive, to add sugar, which is a very popular food source for microbes, to use it as a preservative. In fact, but because of the high concentration of the sugar that you use, or for example, let's take an extreme case of high sugar concentration, honey, right? Honey is a concentrated form of sugar, but it's not easily contaminated with microbes, in fact, because even though lots of sugar is there for the microbe, there isn't much water, right? So salt, concentration of the food material itself, as in the case of jam, which has lots of sugar, and it has, now here we have an example of your cooked strawberry, right? If you don't want fresh strawberry, then you can use heat and turn it into jam and add sugar. Both the heat and high concentration of sugar actually will work as preservatives. Some bacteria, though, is still capable of living in high salt concentrations, as is Staph aureus. So typically, microbes, typical pathogens do not grow in salt solutions beyond 3.5% or so. Okay, Staph aureus can actually, and some Pseudomonas species, but particularly Staph aureus, can survive in solutions of up to 10 or 12% salt. Okay, drying, so dehydration, of course, is also very good because you remove all water without adding any sugar or salt. But again, you can dry anything, okay? So there are some foods that you can enjoy being dried. Pharmaceuticals, drugs, antibiotics, vaccines, those can also be safe to freeze dry, and they can last for a long time. In fact, they have a very extended shelf life. But you don't want to have lyophilized strawberry again, right? In this case, or lyophilized steak. Right. Cases of salmonellosis from dried eggs, of course. Fresh eggs could have salmonella. So freeze drying can preserve bacteria, in fact, as well, some bacteria, right? Because it preserves the material itself. And it stops any form of life. But you can lyophilize bacteria and it will not actually kill them. And here I can give you another example of, say, mycobacteria, right? Mycobacteria, because of its waxy envelope, it's resistant to dehydration. And it can survive, in fact, in the environment, even though it does not produce endospores. It can survive in the environment for months and years. And looks like, ladies and gentlemen, that is the conclusion of our chapter for today. So, any questions? If you're still awake out there, give me a shout that you are all good.
Yeah, we're all good.
Excellent, excellent. And you're still awake. That's good. Right.
Professor, these lectures are being recorded. When are you going to post it on D2L?
Yes, this lecture was recorded, and I will post it later on today or this evening onto D2L. Okay, I'll make a special folder for recorded lectures. Okay.
And another question, do you have any idea if the exam will be in person or will be online,
The midterm exam or the final?
the final?
Oh, it will be online.
How about the midterm? Everything will be online?
Yes, so far, unless the university changes policy.
Okay, okay,
Okay, it's allowing us to carry out the final exam online, unless they change policy and things open up. They may ask us to reschedule for in person. Okay, but
Right.
I'll keep everyone posted.
Just looking for like long-term plans because I'm not in Sudbury right now.
Yes, absolutely. I'll make sure. I mean, if this is were to happen, those who are, I know some students might be even outside of the country, not just outside Sudbury.
Oh, yeah.
But in those cases, I mean, I'll have no choice but to make special arrangements, obviously, with them, right? I will have to make special arrangements. But for those who can, we would expect them to attend if it were to be in person. Good?
Yep.
All right then. So enjoy the sun, even though it's very, very cold. Stay warm, stay safe, and we will talk to you next week. Bye bye.
Professor, have a nice day.
Have a good week.
You as well. You too. Thank you.